Welcome to Study Time, a televised home learning program produced by Rwanda Basic Education Board.
Hello viewers, here we are again, the grade 10s. So, like I just talked about, that this lesson is meant for Senior 5 Sciences. We've been dealing with protein synthesis. We've covered the whole of it. We've talked about amino acids, we've talked about proteins, we've talked about the cell, we've talked about ribosomes, and as you can check, in the background image, we do have a whole process compounded into one image. So all the words that you see there are giving us a whole picture of what we've actually been doing. So interesting. Just one picture and takes it all. It also means that we actually don't need a summary. We don't need a summary here. So, the only thing we need in this lesson is, are we able to answer questions? And if we are in position to answer these questions, how? What do we need? Which words do we need to pay attention to as we do the writing process? What is the examiner assessing? What is he looking for when he sets a question? So setting a question, therefore, means that you as a candidate must read the question, interpret it, and be in position to answer as appropriately as possible. So you attempt, you try to give the most likely acceptable response to the question that you've been given. Some of these questions come with data. Some will have graphs, others will have tables, others are just images, while others require you to think and break down whatever you've been given as a text. Others will come as extended writing in demand, which also means that your writing skill is looked out for. So let's start. You have a question there. Carefully look at it. All I'm going to do is to give you a fair guidance on what to think about when you come across such a question. I'm actually saying there are no answers. I'm very sure you have read the question finally, very well, and you should be in position to start attempting. Some of these things look obvious and pretty simple. One mark, 2, 3. I mean, not a lot is being asked for here. So what do we make of the question? When we look at B, does it remind us something like a 5-carbon or pentose? when we look at A and B, then A coming again and B coming again, does that remind us of the alternating units? And if the question we do have in front of us is about the DNA, then definitely I would not need to start thinking about RNA. As simple as that. It will also be a guidance to me that I can't think of ribo or a ribose sugar. Ribose as a 5-carbon, or deoxyribose also as a 5-carbon. So my guidance, therefore, is just here where the laser is. So let's check what this has for us. Remember that the picture we do have here has to keep reminding us of the topic and the very things we are responding to as questions, so that we do not go astray. So that we do not go astray, as in go off topic. So I'll take you back to the slide again. We look at the question. And then read one part and go to the slide one more time. So each of these will actually give us a guidance on some of the labels. If you can check, for instance, question C. And when we head back to the question one more time, that describe the role of tRNA in translation. Describe the role of tRNA in translation. We got it here, and we move to the response. Which actually is in the picture. Actually, the very statement you see there is in the picture. When we use the word docking, like the way ships, aeroplanes do taxiing, buses park, ships dock. In the same way, tRNAs will dock, fix, attach, okay? It therefore means that the use of these words, though they are English words, we use them to give us some bit of guidance on what we actually mean. Move, fix. So that's the docking. Until a whole protein is made. Now you need to check the process and look at the statements. One in, another one out, until we finally have the whole structure forming up here as our polypeptide chain, the primary structure of the protein. So you have our polypeptide chain forming. It's forming, and this will undergo other processes until we finally have a functional protein. That is the role of the ribosomes during protein synthesis. It also shows the role of the tRNA. Here it is. What it is carrying is added as we keep moving. The other question, a bit lengthy. There is so much important info that you need to read very carefully. And it also shows the application of what we actually learn. So don't tell me, teacher taught us things which do not have any life applications. No. We need to think about genetic disorders. We need to think about how they can be minimized in the body. Since we cannot remove these mutated genes, can we actually lock them, stop them from revealing what they have as wrong information? If we got a chance to do that, then that means that at least we would lessen the expression of that wrong gene. Remember, we talked about gene expression. All I'm trying to say is genetic drugs are actually designed and made in a format resembling part of the information we use to make the proteins, to express the genes. So let's check it out. The first part is saying copy and complete the table. Guidance, complementary base pairing. And you're done. Let's check the second question. A small bit of guidance here. So I want everyone to pay attention here. We need to start thinking. If we say anticodons, and then the codons on the mRNA, anticodons, and then codons. When we talk of codons, we need to think. What is the mRNA? Secondly, what is tRNA? How do they complement? So that if they give me a strand of mRNA, and I'm to create one of tRNA against that mRNA, can I make it? So if we are making proteins and we need transporters for amino acids, the tRNA, it means, therefore, that if I'm to have a drug that is supposed to stop the mRNA message being read as a wrong gene, what does this tell me? Do I need to start thinking about a drug, medicine, a genetic drug that is designed like the one to carry an amino acid, but because that mRNA is wrong, then I have to use a genetic drug that is looking like an tRNA that is going to come and dock? So if it docks, wow, an amino acid should be added to form a protein. But, oh yeah, this is a drug. It's not a true tRNA, so it is not going to be in position to specify a certain specific amino acid to bring and add to the chain. Interesting. So, if this comes and docks, then it means that this information is not going to be read. Probably. I use the word probably. Yeah, so that protein won't form. And if it does, it will be to a smaller extent. Wow, so the medicine is found. Genetic effects are lessened. So with that as a guidance, let's check what would have been the responses. What is supposed to put the complementary bases together, the processes in which we can have a protein, or how the DNA can form a copy of itself, but this time as a single strand, in which we look at it as mRNA, and so we look at that as transcription. And then the bases, definitely. Check the way, try to look at the way they are arranged in groups of 3. Codon. In groups of 3 as a codon. Remember that. C-O-D or N. Codon. C-O-D-O-N. Got it. Now, the other part is the extended writing. Ababobina. No, this time around, you do not cram. You need to interpret the question, get exactly what it requires, and look at how much as a mark they're asking you to score. Essentially, these 2 questions, I love them. Check the stake. 20 marks. The score required here, 20 marks. So if there are 20 marks, then we are looking at 15 marks for the first part of the question, and then 5, as you can see, 15 marks for the first part, 5 marks for the first part, I don't know what's happening, and then 15 marks for the second part. You also need to look at the weight of the question, how much it requires you to do the writing. So the writing process, examiners are going to be looking at do you have the right terminologies? Have you made any errors using these terms? And If so, by how much? What is the damage? Is your writing sequential? Is it continuous writing? Such that one component gives birth to another? I'm trying to say that the flow of your information should be in a way that it is guiding. That you can't start talking about translation before you actually talk about transcription. Then there is no flow. You're not going to fail to talk about transporters for amino acids, and you think that you're making a process of having a protein. What is needed in transcription? Are you able to cite words like cistrons? What about the intruders we looked at some time back? What about the exons? Exons, remember them? EXONS. So if you've read the question very well, they say we need only to define, check, we need only to define transcription and translation, indicating, so we need to tell. We need to be able to show precisely why these processes are actually important in our bodies. What if one is off? Does the other one work? What if it went wrong? What would actually happen? What value do we attach them to? And secondly, explain fully. Remember, in explanation, you give reasons as to why things appear the way they are. So there we go, the first one, like I talked about, the 5 marks. Which components do we need in our answers? Where do we make errors, and how do we correct them? So you need to practice the writing part of it. You need to store a memory in your hand as you write. When you're writing, your hand should have a memory. The same applies to drawing. The more you write the word transcription, the less you're going to have an error in its spellings. Complementary. Specific regions. Or if you can't have that, can you shorten the word cistron? And then what about translation? When we say the importance, a person, when we talk about the importance here, when we talk about the value, we are saying once translation has taken place, once transcription has taken place, what good in it do we benefit? Could they be the proteins? And if they are, what are the examples? Proteins that are meant for blood clotting. Can we mention others? Can we talk about red blood cells for courage? Can we talk about hemoglobin? Can we talk about enzymes? Can we talk about hormones and their value? That's what we are talking about when we say the importance of translation and its value to organism survival. The extended part, again, remember, though we have 15 marks and the text looks shorter, we are looking at your writing, your sequence, the flow. That's where our focus is. We are looking at how you use the terminologies, how you link them, how you describe the process, but also giving a reasoned explanation for each step. So you must have mastered the concept. And you must have a true understanding of what actually happens. Reasons as to why each step is needed. Exactly, that's what we are trying to talk about. And the continued part of it when it comes to translation, like we just talked about. I hope you've taken a snapshot of that so that you're able to read it very carefully in your own time and link it with what the question is asking for. And then you have another small short question here. Again, about tRNA. So simple, very easy. What we have? When you look at part B is actually taking us the very things that we have just finished. And so we are not going to deal so much here because it's like a repeat of what we just looked at. Remember that these questions continuously come in a variety of ways. So it requires your interpretational skills. You know, oh, this is the same question I looked at the other time. So you don't need to cram which answer is for which question, but you need to have a clear understanding. Interpret the figures, interpret the images, and you'll find that it's the same. And like we do have here. Not so much. And as we wrap up, remember, you need to be having your camera close by or a phone. Take it and then be able to refer to the questions before. The last part is an extended question. Research-like based question. I want you to find the link between how DNA makes errors, replication, formation of sickle cells, and global distribution of malaria, and the frequency of these traits. So read the question very carefully. And with that, we come to the end of the session. Thank you so much for following.